Okay, in this special edition lecture for lab, we're going to look at white blood cells, also known as leukocytes. You should know who the white blood cells are. You should know what they do. You should know their population, meaning their numbers, most common to least common. You should know classes of white blood cells or leukocytes. You should know their unique characteristics. So to the right on the screen here, you can see some different white blood cells by name. All of these are leukocytes, but they have their own specific names, and you've probably heard of most or all of these before. First up, we got the neutrophil. Neutrophil. So you got three of them you notice here that have phil in their name. As far as ease and organization, please notice here that they each have granules. Granules are storage containers holding some type of particular chemical substance that they use to do whatever they do as far as killing microorganisms. So the substance in each of these cells, granules, are going to be different in chemistry. And because they're different chemically, that means they stain differently when they're stained with a prepared microbiology or lab stain. Neutrophils stain a pale lavender color. Basophils are called basophils because they take up a blue dye. Okay, in this special edition lecture for lab, the eosinophil white blood takes up the red dye in the right, also known as leukocytes. So you have one cell that becomes more reddish white blood cells are. Appearance in granules. You should know what they do. Becomes more bluish black. You should know their population, meaning their numbers, and one that is pale, most common to least common, and so pale that you can easily see its nucleus. You should know classes of white blood cells or leukocytes. These three are collectively known as the characteristics or granular leukocytes. So to the right on the screen here, on the other side here, you have white blood cells two lymphocytes. All of these are leukocytes, and a much larger monocyte names. These you've probably heard of most or just count before. There are two types here: monocyte, lymphocyte. First up, we got the neutrophil, or agranular. So you got three of them. You notice here the other three on their name, right side of the screen. As far as ease and organization, what's unique about these two, other than being each have granular, is that granules of white blood cells, storage containers holding some type of the smallest of white substance that they use, are known as lymphocytes. And whatever they do, you may have heard of before, killing microorganisms. Lymphocytes are associated with substance in each of these cells. Granules HIV, different. HIV likes to wipe out because they're different chemically. That means they stain differently when they're stained with the prepared microbiology or abbreviations for neutrophil stain. The origin of those lavender particular lymphocytes, whether they're basophils are called basophils because they take up, or whether they originate in rights in the bone marrow, RIGHT cells. The eosinophil. So we're going to continue on and look at these five cells in detail. So you have one cell that becomes more reddish. You notice that they have nuclei and granules. One that becomes more bluish black or purple color. They do not contain, and one that is pale. Hemoglobin is so pale that you can easily see its nucleus, erythrocytes, which is multi-lobed. These three are fewer in number as the phils or granular leukocytes. They take up different colors. On the other side here, you have two lymphocytes side by side. Acid or a much larger monocyte of right stain. These two cells, we just count that there are two types here, monocyte lymphocytes, are agranular in comparison to the other three on the right side of the screen. So when we look at the white blood cells, we've got granular sites and agranular sites. To focus on the granular sites first and foremost, again, there's three cells that end in the word phil, the smallest of white cells. They originate from bone marrow, lymphocytes, and they're all more or less phagocytic. Now, the most phagocytic, and the first answer you should think of when we say a phagocyte, HIV, which means HIV, like cell that eats certain populations, is known as the neutrophil, which happens to be the most common. T and B cells are abbreviations for white blood cell in circulation. The origin of those particular lymphocytes, whether they're educated and originate in the thymus, histolaboratory, they originate, and because they have a multiple lobe nucleus, they're called polymorphic nuclei. So we're going to continue on and look at those neutrophils. You can see detail. Four of them here. On the left of the screen, you notice that they have nuclei, and the most numerous cells they do not see in this field here. Hemoglobin, or your erythrocytes, are red blood cells. The white space in between cells is liquid plasma. Remember, plasma They're fewer in number is matrix and larger. And from histology, you should remember what matrix is. They take up different colored stain, and as they do, they take up the blood as a liquid acid or the basic component of connective tissue. Anyways, back to the neutrophils. They're a pale lavender in color with multiple lobes to their nucleus, and you can see the different shapes here. They're never going to look the same as far as the position and pattern. So when we look at the white blood cells, which you got granulocytes and agranulocytes. To focus on the granulocytes first and foremost, when a neutrophil or any of the granulocytes for that matter, they originate from bone marrow, and they're all more or less phagocytic. Now the most phagocytic, and the first when they're young, they're referred to as bands in histology, lab reports, cell that eats. And when they're mature, they're known as the neutrophil, which happens to be the most common nucleus, SEGS. So if you see in white blood cell in circulation, segs and bands, that's what that means. They're also referred to as PMNs, white blood cells, in someone's histo lab report, because they have a multiple lobe nucleus. The fine granules of neutrophils, which are pale lavender in color, carry various germicides, such as peroxide, 4 of them here, leech-like substance. The small purple flecks are platelets, and the most numerous cells that you see in this field here, H2O2 is erythrocytes or red blood hydrogen cells, peroxide, the white space in between cells, while we're dealing with liquid plasma as well. Remember, plasma, in addition to those germicides, is matrix. And from histology, you should remember what matrix is. Primarily destroy bacteria. Is a protein. The blood is a liquid connective tissue, as fluid connective tissue. And defensin is used essentially to poke holes and damage. They're pale lavender in color with multiple lobes to their nucleus, and you can see the different shapes here. They're never going to look the same as far as the white blood position and pattern of their nucleus. Next up, we have the eosinophil, which I had mentioned. So there is a reddish-orange color. Neutrophil or any of the phils, the granulocytes, they're much rarer than the neutrophil. Are young. Neutrophils, eosinophils, and basophils, they destroy parasites. When they're young, they're referred to as bands through releasing histology enzymatic substances. And when they're mature, they're going to be referred to as substances that are found segmented within eosinophils granules. So if you see in someone's blood report, segs and bands, they primarily young, kill and destroy parasites. Immature parasites are generally larger than bacteria. So what essentially happens is you get fine granules of neutrophils, which are pale lavender in color, of some sort, carry various germicides, such as peroxide, disease-causing leech-like substance agent. So that's what this abbreviation is. Who the common parasites are, as far as chemical formulas go. H2O2 is peroxide. They also play a role in gobbling up. We're dealing with the bleach substance, phagocytizing. In addition to those germicides, allergic substances or allergens, bactericides, because neutrophils allergen antibody destroy bacteria. So the AG is a protein antigen spear. The small AB is for antibody. And defensin is used essentially to something foreign, damage your body does not agree with. Fungi. Your immune system responds by producing neutrophils, antibodies. Most numerous white blood antibody in your blood to neutralize. Next up, we have the eosinophil, which I had mentioned, potentially harmful. It is a reddish-orange color in appearance. You can see one here in this picture. They're much rarer than the Lastly, neutrophil. For the 3 granulocytes, the rarest of white blood cells is known as basophil. And they do so picture here, which is an illustration. You can see that there are several down at the bottom here, blue granules, but they can be found purple or almost a dark black eosinophils granules. How long they've been stained. And those granules interfere with our ability to parasites, make out the nucleus. Parasites are generally larger. The nucleus is usually a U or an S shape. What essentially happens is you get several. Usually has 1 or 2 lobes, or chewing on some 2 lobes to it, or some parasitic. And it's blue in appearance and color because of causing the basic or basophilic blue dye. Who the common parasites are that an eosinophil. Basophils play a role with allergies, producing 2 substances that you may have heard of. They also play a role in gobbling up, known as histamine, phagocytizing, which causes inflammation or promotes leakiness. Allergic substances or allergens, known as call it a vasodilating allergen antibody complex. That also attracts white blood cells. AG is an abbreviation for antigen. We also have the other substance that's for antibody released by basophils. When you're exposed to something foreign that your body does not agree with, your immune system, which means responds by producing antibodies. And the purpose of an antibody in labs, laboratory settings, the hospital, you'll notice that the glassware that comes in contact with potentially harmful, say that it's heparin coated. because the glass will cause a clot. Lastly, for the three granulocytes, the rarest of white blood cells is known as a basophil-like substances. And in this picture here, which is the very bottom here, you'll notice that there's a cell under the line. In this case, blue granules, but they can be blue, purple, or almost dark black, even color, related or similar cells and function to basophils. Only they're found in connective granules interfere with our ability to they also produce nucleus. Histamine. The nucleus is usually a U or an S shape. The urticaria or hives that usually has one or two lobes, or experience has two lobes to it. You're exposed. Again, to some blue in appearance and color because of the irritating substance on your skin. The basic or basophilic blue dye. So those are your three granulocytes. Basophils play a role with allergies, again, are referred to as agranulocytes because they don't have known as histamine, plasmic granules, which cause at least inflammation, visible, promotes leakiness of blood vessels. And this happens to be the largest of white blood cells known as monocytes. That also attracts white blood cells. And the smallest of white blood cells have the other substance that's stored as lymphocytes, released by basophils into circulation, known as heparin. Lymphocytes are the second most abundant anticoagulant, which means it interferes. So you're going to need to know the 5 major white blood cells. And most common to least common settings, the hospital, modify that list. You'll notice that what glassware that comes in contact with blood, the least common granulocyte, heparin coated. The slide on the screen says because the glass you should cause lymphocytes are more common than monocytes. And to prevent clotting, they coat glassware with heparin-like. They're small. You can see almost the very bottom here. You'll notice there's a cell, red blood cell, under the line, side by side, referred to as your typical lymphocyte. Is the smallest cells are related or similar cells and function. Small cells with very large round nucleuses. Notice there's very little cytoplasm. They also produce and release histamine and heparin, and we're responsible for urticaria. You should know that these are the cells that provide you with immunity, experience, memory. You're exposed to some chapter that your immune system has memory or irritating substance on certain cells that are carried in your bloodstream. So those are your 3 granulocytes. The agranulocytes, again, are referred to as lymphocytes are because the cells that are responsible for recognizing plasmic granules, or they're at least activating visible phagocytes. And this happens to be the largest of white blood cells, known as monocytes. More about lymphocytes. There's 2 classes of white blood cells, typically known as lymphocytes. There's 3 classes. There's actually more than that. And lymphocytes are the second most abundant T cells, and you have several types of T cells, which you're going to need to know the 5 major white blood cells, at the end of who's most common to least common. You modify that list. Know that there are 3 types of common granulocytes, T cells, B cells, least common granulocyte, and your atypical lymphocyte known as agranulous. You should know lymphocytes are more common than monocytes. T cells are called T cells or T lymphocytes because they are programmed and educated in the thymus. You can see almost B cells are called B cells because they originate and mature. Your typical lymphocyte is the smallest white cell. Your T cells directly attack cells with very large round nucleus, viral infection, very little cytoplasm. They also destroy cancer cells, tumor cells in the body. And you should know that these are the cells that provide B cells immunity, indirectly protect you by producing antibodies. Antibodies are proteins created by your immune system, and it's due to certain in response to an antigen, your bloodstream, known as. And lastly, the natural killer. I say that they're atypical. They're atypical in that they're very large cells that are responsible for recognizes your typical lymphocyte is very small with activating large round nucleus phagocytes and agranular. Natural killers kill cancer cells. Natural killers kill whatever they don't recognize. There's 2 classes. There's actually more than that. So they're also a killer. There's 3 classes. The T cell at the very top has several populations of T cells. One of them are going to be called cytotoxic T cells, which I'm going to say are killers. As well, lecture at the end of the semester. We don't call them a killer, instead we call them cytotoxic. For now, you'll just know that there are 3 types of lymphocytes. The T cell and the natural killer are atypical lymphocyte, involved in different aspects of your immune system. T cell is referred to as called T cells, one is T lymphocytes because they are programmed and educated in a short little mini lecture of lymphocytes. B cells are called B cells because they originate in an mature immune system, the marrow. Versus the T cells are part of your T cells directly attack system, and some invading more about that at the end viral infection or throughout the semester. Viruses, as they also destroy cancer cells. The largest of white blood cells is known as a monocyte. Your B cells
Monocytes are very large cells
protect you by producing antibodies.
Irregular
Antibodies are proteins created by your immune system,
usually a U-shaped
in
nucleus
response to an antigen.
They appear a foamy gray color or
And lastly,
pale
the natural
purple,
killer. I
depending
say
on
that
the
they're
stain
atypical.
and the staining
They're
procedure.
atypical in that they're
You
very
should know that
large,
monocyte is the
and
term we
granular.
use when it's in circulation, and when
lymphocyte
that
is very
white blood
small,
cell
with a very
exits
large
the bloodstream
round
and
nucleus
enters tissues and
and
organs in
agranular.
your body.
We call them
Natural
macrophages.
killers kill.
So a monocyte
Cancer
and a
cells,
macrophage
natural
are essentially
killers
the
kill
same cell,
whatever
it's
they
just
don't
recognize.
name something different.
So they're also
based on
a
its
killer.
location.
When they lie
the T cell
within
at the very
different
top
organs, they're
has
called
several
resident
populations.
macrophages.
One of them are going to be
And
called
examples at the bottom of the
cytotoxic T
screen
cells.
here would be
They're
Kupffer
killers
cells,
as well,
which are
but they
found
we
in
don't
your
call
liver.
them killer. Instead, we call them
We have
cytotoxic
dust
T cells,
cells,
which would also be
the
called
T cell and the
alveolar,
natural
found in the
killer
lungs.
are
And we
involved
have
with different aspects of your immune
Langerhans cells, which
system.
are found within the skin. One is referred to as innate
And
immunity,
lastly,
and
we
one is
got
referred to
microglial
as
cells, and
adaptive
you might remember
immunity.
from part
And
one
while
that
I'm
those
on
were
this
found in your
tangent in
brain.
this
So
short
these
little
are
mini
all
lecture of
phagocytic type
lymphocytes,
cells,
you should know that the natural
macrophages.
killers are part of your innate immune system.
And monocyte
Versus
in
the
circulation
T cells
within
are
the
part of your
bloodstream.
adaptive. It's referred to as a
immune system, and I'll
macrophage
tell you
when
more
it's
about that
surrounding
at the
tissues
semester.
and organs
Outside
or
of the
throughout the
bloodstream.
semester as needed.
Both are phagocytic.
The largest
both
of
are
white blood
involved
cells
with
is known as a
fighting
monocyte.
chronic infection and inflammation.
Monocytes are very large cells with
Phagocytosis from part one
irregular
anatomy.
nucleus.
You should remember.
Usually a U-shaped
Are
nucleus. They
involved
appear a
with
foamy
eating and
gray
destroying
color or a
foreign
pale purple
organisms,
depending on the
dead and
stain
dying
and the staining
cells.
procedure.
And there's
You
going to
should
be a
know that
relationship
monocyte
that you
is the
learn at
term we
end of the
use when it's
semester
in circulation,
between
and when that
large
white blood cell
phagocytes
exits the
and
bloodstream and enters tissues and organs in your body.
Lymphocytes actually help
Program some
macrophages.
types of white blood cells.
So a monocyte and
as
a
into
macrophage are
what to
essentially
look
the same
for and
cell.
what
It's
to
just
eat
and kill
name something
or
different
destroy.
based on its location. When they lie within different organs, they're called resident
So a little more on
macrophages.
the whole immune
And
role
examples
with
at the
white
bottom
blood
of the screen
cells.
here would be Kupffer
Their
cells,
function,
which are
the
found
lymphocytes
in
for
your
that
liver,
matter,
produce
have
antibodies.
dust
The
cells,
purpose of antibodies
which
are
would
to
also
help
be called
inactivate alveolar
found in the lungs,
antigens, and we have
and to help
Langerhans cells, which
program
are found
and
within the
awaken
skin. Phagocytes, and
as
lastly,
into
we
who to
got
kill.
microglial cells, and you might remember from part one that those were found in your brain.
So
these
what's
are all
unique about
phagocytic
this list
type
of
cells.
phagocytes is
Macrophages
neutrophils
are phagocytic, but they also kill by releasing
monocyte,
toxic
if it's in
substances
circulation
to within the bloodstream.
bacteria.
It's referred to as
That
a
was that bleach
macrophage
and
when
that
it's in the
peroxide-like
surrounding tissues
substances
and organs,
outside
or the
of the
defense
bloodstream.
and protein-like spear that it uses to damage
Both are
the membrane.
phagocytic.
Both are involved with
Victim.
fighting chronic infection and
On the
inflammation.
screen,
the word agglutination is a fancy
Phagocytosis
term
from
for
part one
clumping,
anatomy.
a clumping
All of you
event
should
that
remember.
occurs that I'll discuss at greater length when I
Are
review blood typing with you.
involved with eating and destroying
Some clinical
foreign
information related to white blood
organisms,
cells
dead and dying
would be
cells.
to know terminology, and there's
Like
going to
the
be a
words
relationship that you
leukopenia
learn at the end of the
versus
semester
leukocytosis.
between large
Leukopenia is
phagocytes
lower than normal
and white blood cell
lymphocytes.
numbers,
The
while
lymphocytes actually help
leukocytosis is
program
above
some
normal types of
values.
white blood cells.
Notice
Notice there is
into
a range
what
here
to look for
between
and what to eat to
5,000
kill
and 10,000
or destroy.
would be the normal range of white blood cells per cubic millimeter of
So
blood.
a little more on the whole immune role
Or
with
white blood cells,
liter.
their function, the
You'll notice a table
lymphocytes
in
for
your
that
textbook
matter
that you should
produce
review and know
antibodies.
well.
The purpose of antibodies are
explains
to
and
help inactivate
outlines the textbook's
antigens
descriptions of
white blood cells,
and to
red
help
blood cells, and
program
platelets.
and
Some more
awaken
terminology that you should work on and
phagocytes
know
as into who to
relate
kill.
to different given populations of white blood cells. We've got two
So
terms
what's unique
that relate
about
to
this list of
lymphocytes. We've got two
phagocytes
terms
is
that relate to the
neutrophils
monocytes
are phagocytic,
two
but they
terms
also
for the
kill by
neutrophils, two
releasing
terms
toxic
for the
substances
eosinophils,
to
and two
bacteria.
terms for the basophils. That was that bleach and that
Leukemia,
peroxide-like
which all of you have heard
substances.
of before,
Or the defense
is a
and
cancerous
protein-like
condition of
spear that it
blood,
uses to damage
specifically
the
related
membrane
to
of its the formation
victim.
of abnormal cells.
On the screen, the word
So I've given you
agglutination
two different
is a
descriptions
fancy
of the
term
word
for
leukemia, a clumping event that
progressive,
occurs that
meaning
I'll discuss
chronic,
at greater length when I review
advancement
blood typing with
abnormal, non-functional.
some clinical
leukocytes.
information related to white blood cells.
Leukemia
Would
originates
be to know
from
terminology
blood-forming stem
like the
cells
words
found in your
leukopenia
marrow
versus
Marrow
leukocytosis
is
found in bone, specifically the
Leukopenia
spongy,
is lower than normal
red
bone marrow
white blood cell
location.
numbers.
while leukocytosis
There are
is
different
above
forms of
normal
leukemia
values
that you may have heard of
Notice
before.
there is a
Acute
range
versus
here
chronic
between 5,000 and
myeloblastic
10,000.
versus lymphoblastic
Has to do with
would be the normal
certain
range of white blood
populations of
cells
white
per
blood cells.
cubic millimeter of blood.
You should know what a differential blood cell
or
white blood cell count
microliter.
is, what the normal ranges are.
You'll notice a table in your
When
textbook that you
populations
should
change,
review
you might
and
think
know
of
well.
why is there an
That
increase in the
explains
number
and
of neutrophils?
outlines the
Why is there a decrease in the
textbook's
number of
descriptions of
monocytes? It might be
white blood cells,
related
red
to
blood
the
cells, and
number of
platelets.
some more
Or the
terminology
type of
that you should
infection
work
that
on
you
and
might
know
be fighting off.
relate to different given populations of white blood cells. We've got two terms that relate to lymphocytes. We've got two terms that relate to the monocytes, two terms for the neutrophils,
And this
two terms
concludes-
for the eosinophils, and
not
two
yet.
terms for the basophils.
But
One more point that I want to
which all
make,
of you have
one
heard
or
of
two
before,
more points that I want
is
to
a
make
cancerous
about white
condition
blood
of
cells.
blood.
So you know that
Specifically
we
related
just looked
to
at 5
the
unique types
formation
of
white blood cells.
abnormal cells. The rhyme at the top of the screen might
So
be
I've given you
useful in
two
helping
different
to
descriptions
memorize
of the word
who's
leukemia.
the most common to least
A
common and what
progressive,
are their
meaning
relative numbers in
chronic.
percent. This is
Advancement
60-38-30
of abnormal,
as in percents.
non-functional leukocytes.
Never let my engine blow. The first letter of each
Leukemia
word
originates from
stands
blood-forming
for
stem
neutrophil,
cells
lymphocyte,
found in your
monocyte,
marrow.
eosinophil,
Marrow
and
is found in
basophil.
bones.
You can see a distribution of white blood
red
cells
bone
of
marrow
who's the most
locations.
common, who's the least common.
There are different forms of leukemia that you may have
All
heard
of your white
before.
blood cells
Acute
lie
versus
within
chronic.
a component of a
myeloblastic
blood
versus
sample known as a
lymphoblastic
hematocrit.
with
The
certain
hematocrit and
populations
the
of
packed
white
cell
blood
volume
cells.
relate mostly to the red
You
blood
should know
cells
what a
that you see
differential
listed at the
blood
bottom
cell
of this
white
tube.
blood cell count is.
The white blood cells lie
the
within
normal
this
ranges
white
are.
band known as a
When
Buffy code,
populations
which
change,
makes
you
up
might think of
less than 1%. Why is there an
total
increase
blood.
in the number of neutrophils?
Why is blood a
Why is there a
connective tissue?
decrease
This
in
will
the
be
number of
repeated more in
monocytes?
lecture.
It might be
because it's
related
mostly
to
plasma.
the number
There's a
of
55 to 45%
or the
ratio
type of infection that you
non-living
might be fighting
to the
off.
white band down. You're looking at about 45% of a sample of blood being living cells, or collectively known as formed elements. And this concludes
The development of white blood
No,
cells
not yet.
originates from one more
The
point that I
marrow in
want to
your
make,
bones.
one or two more points that I want to make
Hematopoiesis is
about
the
white
fancy
blood
term
cells.
to form
So you know
blood
that
cells.
we just looked at 5 unique
Leukopoiesis would
types
be the
of
specific
white blood
term
cells.
for forming
The rhyme at the top
white
of the
blood
screen
cells.
might be useful in helping to
All
memorize
blood cells
who's the most
originate
common
from
to
a
least common and what are their
blood-forming stem cell
relative
called a
numbers in percent.
hemocytoblast.
This is
Which you can see in
60-38-30
this
as in
small
percents.
written
Never let my
table here,
engine blow. The
also
first
found in
letter
your
of
textbook.
each word
The
stands for
hemocytoblast is a
neutrophil, lymphocyte,
blood-forming stem
monocyte,
cell.
eosinophil,
Okay.
and
It
basophil.
originates
You can see a
from the bone
distribution of
marrow,
white blood cells of
creating
who's the
two
most
different
common,
types of stem
who's the
cells
least
known
common.
as progenitors. One is referred to as myeloid.
All of your white blood
one is
cells
referred
lie
to as
within a
lymphoid.
component of
that I
a
just
blood
mentioned
sample
that there are
known as a
different types of
hematocrit.
leukemia.
The
Myeloid
hematocrit and the packed
and
cell
lymphoid
volume relate
types of
mostly to
leukemia.
the red blood
So
cells
you
that
would
you
get
see
an
listed
idea
at
of
the
what
bottom of this
population
tube.
of cells would be
The
involved.
white
in a
blood
given
cells
type
lie
of
within
leukemia
this white band known
scenario,
as a Buffy code.
which
I
makes
start
up
on the short
less than
list here
1% of
first.
total blood.
Lymphoid stem cells
Why is blood
or
a connective
progenitors
tissue?
develop
This will be
into
repeated more in
lymphocytes.
lecture.
Because
Young,
it's mostly
lymphocyte
plasma,
is known as
there's
a
a
55 to
lymphoblast.
45%
Eventually, you
ratio
can
of
see there are different
non-living
populations of
to the white band
lymphocytes
down. You're
mentioned at
looking
the
at
bottom of
about
this screen.
45%.
On
A
the other
sample of
side
blood
here, you
being
have myeloid
living
stem
cells,
cells,
or
which
collectively
originate
known as
from
formed
the
elements.
original blood-forming hemocytoblast. The
but
development
become
of
the
white
various
blood cells
fils and
originates from
monocytes.
the marrow in your bones.
Neutrophils.
Hematopoiesis is the fancy term to
over here.
form these
Blood
two
cells.
areas, or
Lines we'll worry
leukopoiesis
about
would
in
be the
lecture.
specific term for.
We've
We've got
forming
basophils,
blood
neutrophils,
cells.
eosinophils. All blood cells
And
originate
monocytes.
from a blood-forming
They all
stem
originate
cell
from
called a
myeloblast,
hemocytoblast, which
which
originated
you
from
can see in
myeloid
this
stem cells,
small
which originated
written
from
table here,
also
also
the
found in your
hemocytoblast
textbook.
and bone marrow. The hemocytoblast is a blood-forming stem cell. Okay, it originates from the bone marrow, creating two different types of stem cells known
The
as
number you
progenitors.
should put to memory
One is referred
for
to
the
as range
myeloid,
of white blood
one is
cells
referred
is
to as
4,800
lymphoid.
to
I would
10,000.
remind you that I
800
just mentioned that there are different types
cubic
of
millimeters
leukemia, or
myeloid,
microliters
and
of
lymphoid
blood types of leukemia. So you would get an idea
And
of
this
what
will conclude
population
your
of cells
blood
would be
lecture.
involved in a given type of leukemia scenario.
If I start on the short list here first, lymphoid stem cells or progenitors develop into lymphocytes. A young lymphocyte is known as a lymphoblast.
Eventually you can see there are different populations of lymphocytes mentioned at the bottom of this screen. On the other side here you have myeloid stem cells, which originate from the original blood-forming hemocytoblast but become the various fils and monocytes. Neutrophils, we'll start over here. These two areas or lines we'll worry about in lecture. We've got basophils, neutrophils, eosinophils, and monocytes. They all originate from myeloblasts, which originated from myeloid stem cells, which originated from the hemocytoblast in bone marrow. The number you should put to memory for the range of white blood cells is 4,800 to 10,800 cubic millimeters or microliters of blood. And this will conclude your blood lecture.